Hello, I'm Dr. Lori Snyder. I am the author of Bacterial Genetics and Genomics. My book has been launched during the COVID-19 outbreak. As a result, we haven't had any book launch events, so I've decided to record some videos to introduce you to the book, and hopefully this will tell you a bit more about the book and what the features are of the book and a bit of what is inside of the book as well. So this textbook, as you can see, is quite substantial, and what we've got inside are a lot of features that will tell you about bacterial genetics and genomics and walk you through learning the topic of bacterial genetics and genomics. The book is divided into 21 chapters, and each chapter is in 7 sections. And as you can see on the spine, the 7 sections are color-coded, so you can quite easily find which section you are in as you walk your way through the book. In the table of contents, it will tell you each of the 7 sections. So part 1 is DNA, genes, and genomes. Part 2 is RNA, transcriptional regulation, and transcriptomes. Part 3, proteins, structures, and proteomes. Part 4, genetics, genomics, and bioinformatics. Part 5, bacterial response, adaptation, and evolution. Part 6, gene analysis, genome analysis, and laboratory techniques. And this takes actually the theory of what you've been learning throughout the rest of the book and applies it. So it gives you kind of hands-on ways to apply the knowledge that you've been learning throughout the book. So the first 2 chapters are very much computational, things you can do on your computer to analyze genes and genomes. And the third takes those things into the laboratory. And then part 7, applications of bacterial genetics and genomics. At the back of the book, we've got a unique feature. Not only is there a glossary, which most books will have, but we have a glossary of bacterial species. So as you're reading through the book, if you come across a bacterial species you're not familiar with, I have tried to include a little bit about the species within the context of what you'll be reading in the book, but also at the back there's a glossary of these bacterial species which tells you what they do, whether they're gram-negative or gram-positive, a little bit more about those bacterial species there in the bacterial species glossary at the back of the book. Also at the back we've got the index, traditional index at the back, so that you can find specific topics throughout the book that you might be interested in doing some more reading on outside of the chapter you might be looking at the time. Okay. Throughout the book we've got some great illustrations. So the illustrator took my pencil drawings and created just wonderful full-color illustrations throughout the book, as you'll be able to see. So we've got illustrations such as these all throughout inside the book. And at the end of every chapter there are a list of key points. So these points will be the key messages throughout the chapter that I'm hoping that you'll take away from the chapter so that you can refresh what you've been learning, what you've been reading about in the chapter. And then there are 3 different ways to test your knowledge in the chapter. The first are the key terms. So these are glossary terms that were introduced in that chapter. And if you go online to our online resources for the book, we've got flashcards set up. So you can test yourself on each of these key terms using the online flashcards, which I think is just a great resource that's been set up by the publishers at CRC Press, Garland Science, Taylor Francis, to be able to test your knowledge on those glossary terms. The second set of questions are some self-study questions. These are kind of short answer questions, short questions that will directly relate to things that are in the chapter that will test your knowledge and your retention of what you've been reading and learning about in the chapter. And the final section, these are discussion topics. So as I say, these topics are presented for discussion in study groups, as part of class discussion, or on your own. These questions go beyond what is directly covered in this part of the book. So you're not going to find the direct answers to these in the book. This is for your independent learning to expand on what you've been learning on in the book, to bring in research articles and things that have been currently happening, things that are expanded outside of the scope of what I could cover just inside of this book. Use the research literature and other reading to explore these topics in more depth. Tips to help prepare for these topics are available online. And so there's some more tips, some more in-depth ideas on how to explore these topics on the online material directly related to the book, and I've also been writing a blog on bacterial genetics and genomics, where each month I'm picking one of these discussion topics and looking at a paper, looking at a topic that relates to one of these discussion questions, and there are 3 of them for each of the chapters, and exploring that, discussing that, blogging about that myself to give you a jumping off point for your own investigations, your own discussions, whether you're going to use that within a study group and discussion group, subject of a debate, however you decide to use that for your own learning. And I would hope that people would look at the blog, interact with the blog, email me, reply on the blog, however you'd like. Okay, so those are some of the features within the book. I do hope that you check it out. At the back we've got a short summary, and it says our understanding of bacterial genetics has progressed as the genomics field has advanced. Genetics and genomics complement and influence each other. They are inseparable. Under the novel insights from genetics and genomics, once believed borders in biology start to fade. Biological knowledge of the bacterial world is being viewed under a new light, and concepts are being redefined. Species are difficult to delimit, and relationships within and between groups of bacteria. The whole concept of a tree of life is hotly debated when dealing with bacteria. The DNA within bacterial cells contains a variety of features and signals that influence the diversity of the microbial world. This text assumes readers have some knowledge of genetics and microbiology, but acknowledges that it can be varied. Therefore, the book includes all of the information that readers need to know in order to understand its advanced material. And for that reason, from Chapter 1 all the way through to the end will give you the information that you'll need to know to be able to understand the topic. I think it is useful for undergraduate students, graduate students, postgraduates. I think it's even a great resource if you're a PhD student and looking for background material, looking to reinforce your knowledge. There'll be information in here for topics that maybe you haven't explored before or that you want to be able to apply to your research. So I hope you check out the book, and I hope you have enjoyed this video. Please do like and subscribe to my channel. I will be posting some more videos about the book, and if you have any questions, leave them in the comments, and I'll see about responding to those comments, and maybe they'll end up as the topic of a future video.